some other aspects of this topic is interested for your students, we can continue this course or kind of discussion with you and your students in next time.
Yes, why not, sir? They can contact you further through mail also if they have any type of query. So that's not a major issue, sir. So, sir, can you give me 2 minutes to call my director or principal, sir? Okay. Hello, sir. So, sir, he will join us within the lecture. So you can start because this lecture will be recorded, and for our student, we will provide this lecture. So just you can start, sir. Please share your slides. Yes. Hello, sir.
Hello. Welcome, sir. Welcome. Myself, Dr. G.D. Gupta. Sir.
Am I audible, sir?
Yes, yes, yes.
Sir, Dr. Pal, am I audible? I think he is disconnected.
He is present, but I feel something is the problem.
Yeah.
Hello. Dr. Paul. Hello.
I think he will join further.
Again, no problem, no problem.
Okay, I'm back again.
Hello.
Hello.
I'm sorry.
Welcome, sir. Welcome.
Me too. Thank you so much for the invitation. And can I start my lecture now?
Yes, yes, please. Yes,
Okay.
please.
Yes. Just a minute. I opened the. Yeah.
It's okay, sir. No problem.
Just a minute, sirs. I just.
It's okay. It's okay, sir.
Okay. Okay, let's see the lecture. I'm sorry, I have some troubles in uploading. I mean, just opening the. Sorry. Can you please? Oh, which one? Okay. And now is what? Oh, this one. Excuse me. Can you hear me?
Yes,
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 sir.
sir.
I
Yes,
can,
sir.
I can.
Okay. So can I go ahead?
Yes,
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 sir.
sir. Please.
Thank you. So welcome to everybody to this short seminar whose aim is to introduce metabolism of xenobiotics. It should be really a short introduction. One of the main research field of our institute, as you can see from the pictures, we are investigating metabolism of some selected substances using animal model and in 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 the department we are investigating some non-steroid anti-inflammatory drugs, capsaicins, and a class of natural compounds called chalcones. But let's start with Hungary. You might have heard about Hungary, and Pécs is a relatively small city in the southwest, sometimes here, part of Hungary, which is close to the Croatian border, and the country itself has got 10 million, about 10 million inhabitants, and Pécs itself, which is a university city, has got about 150,000 inhabitants with a full university. As for myself, I graduated as a pharmacist long time ago, and just to introduce myself, I was up to my age of 65 the head of the department of Institute of Pharmaceutical Chemistry. Present, I'm a professor here. I served the Faculty of Pharmacy as a dean of over 3.5 years, and since 2019 I serve as an editor-in-chief with Journal of Pharmaceutical and Biopharmaceutical Research. So briefly, that's my main achievements so far, and let's turn to the present, the topic of the present day lecture, which is the metabolism of foreign substances. Among them, obviously, drugs are our main interest. This is a picture which shows you the very simplified, however informative fate of a drug which gets into the body. After absorption, the plasma carries the blood flow carries the stuff to almost all each cells of the body, and in some particular tissues, in some particular organs where the so-called receptors are expressed, the biological response, the pharmacological effect is manifested. As for the fate, the other fate of the compounds is elimination. Elimination of the foreign substances from the body can take place in its original form or in transformed, chemically transformed form, and the pathways over which this chemical transformation of the parent compounds occurs is called metabolism. In wider respect, we frequently recall it as biotransformation. So we are mainly interested in this part. I mean, in the metabolic feature of some selected foreign substances. Metabolism of foreign substances, I mean discovery and continuous investigation of foreign substances, is dated back to the middle of the 19th century. The first two publications were published by German researchers, and it was Ure and Keller who discovered the glycine conjugation of benzoic acid. The story of the investigation is very interesting, since the conjugate, the glycine conjugate of benzoic acid, the trivial name of hippuric acid, is isolated from the urine of themselves as a result of self-experiments. Probably today this cannot be performed. However, at that time the regulations were somewhat different than nowadays. Okay, so the first book which summarized the metabolic feature of foreign substances was published in 1947, which was the first issue, and the second edition of the very much the same book, written by Tackwin Williams, was published in 1959. So it was Dr. Williams who coined the terms phase 1 and phase 2 metabolic transformations of foreign substances, xenobiotics, and the phase 1 transformation were classified as oxidation reactions, reduction reactions, and hydrolytic reactions. The phase 2 reactions were commonly termed as synthetic transformations. If we have a look at an overview of these reactions, I mean phase 1 and phase 2 reactions, just to concentrating on the most important details, we can mention oxidation, reduction, and hydrolytic reactions among the phase 1 reactions, and gluconic acid conjugation, sulfate conjugation, acetylation, methylation, conjugation with glutathione, and the conjugation with amino acids. Among others, glycine is a historical issue. The main characteristics of the phase 1 and phase transformations from chemical point of view is that the phase 1 reactions are typically introduce new polar functional groups, increasing the hydrophilicity, water solubility of the parent compounds. And as for the phase 2, with few exceptions, the phase 2 transformations makes the parent substance or the phase 1 metabolites of them even more hydrophilic by means of conjugation of these rather hydrophilic internal molecules, endogenous molecules like gluconic acid, sulfates, and glutathione, and a series of amino acids. There are two exceptions from this general approach, namely acetylation and methylation. These two transformations make the substances, the substrates, less polar than the phase 1 metabolites or the part 1. I mean the phase 1 metabolites of them. Now, which are the main sites? The main sites of drug metabolism is considered to be the liver. In liver, a variety of metabolic enzymes are expressed, typically in very high level. And as we know, it is the liver through which the drugs, the Peros drugs, reaches the central circulation. So each drug absorbed from the intestinal system passes through the liver, and at which site a substantial amount, more or less amount of the original substance undergoes metabolic transformation. There are some other sites of drug metabolism. Among them, the intestine, the small intestine, is also a very important place. However, we can mention the lung, we can mention the central nervous system, the brain in particular, and some other dedicated places. Let's see what are the substrates. What substances can undergo phase 1 and what substances can undergo phase 2 conjugations. As for oxidation reactions, which are the far the most important phase 1 transformation reactions, the substrates are typically aromatic compounds or aromatic moieties of larger molecules, the double bonds, benzylic and allylic carbon atoms, carbon heteroatom bonds, and oxidation of alcohols and aldehydes, also important transformations from this aspect. In the next few slides, I will show you some examples of these oxidation reactions. So this is the pathway through which the Peros drug gets into the central circulation, showing, expressing the importance of liver as a third or the second site of metabolic transformation of drugs, Peros applied drugs. Further, most important enzymic systems are the so-called cytochrome P450 enzymes, which can oxidize a variety of organic compounds. The cytochrome P450 systems are hemp proteins, and they activate oxygen through binding the oxygen molecule to the iron 3 oxidation state of the central iron ion, and the substrate binding site of the protein is typically a very hydrophobic region of the protein. So that's why typically the hydrophobic compounds are bound to these enzymes, and as a result of the active oxygen activation, one of the oxygen molecules is incorporated into the substrate. The other oxygen molecule of the other oxygen atom of the oxygen molecule is converted into water. Just briefly introducing the nomenclature of the cytochrome P450 systems, since most of the brochures describing the drugs nowadays are mentioning those isoforms, those forms of the cytochrome P450 enzymes, which can oxidize the particular drugs. Classification of the cytochrome P450 enzymes is based on the protein homology of the enzymes, and those enzymes which shows higher than 40% homology in their protein structure are falls into one family. Within one family, those whose homology is higher than 55% form a subfamily, and inside the subfamily, we can identify the isoform of the particular enzyme. Nowadays, recently, in the last couple of 10 years, the formation or existence of the alleles, the different alleles of the isoforms, are getting more and more important, since typically these derivatives are showing typically less activity, lower activity than the native enzymes, which obviously affect the pharmacokinetics of the drugs applied. No, so this is the cytochrome P450 cycle. I do not want to get into details, just to mention some important steps which can help to understand the conversions, the chemistry of the metabolic transformations. So in the resting state, the iron oxidation number of iron in the cytochrome P450 is iron 3 plus. In this resting form, the substrate, the sixth substrate binding site of the iron is water. When the substrate, I mean the xenobiotic, binds to the protein part of the binding site of the cytochrome P450 enzyme, okay, due to the change in the structure, the three-dimensional structure of the hem part, the redox potential of the iron 2, iron 3 system are changing somewhat, and as a result, the first electron is transferred to the iron part of the hem. So the iron 3 is reduced to iron 2, and the name of the enzymes is originated from the characteristics. One of the important characteristics of this substrate-bound iron 2-centered cytochrome P450 enzymes, namely in vitro experiments, this form of the enzymes can bind carbon monoxide, and the carbon monoxide complex of the cytochrome P450 enzymes displays a very intense absorption band around 450 nanometer. That's the origin of their name. So in the next step of the cycle, molecular oxygen is bound to the iron 2 center, replacing the water, and afterwards a second electron is transferred to the complex. The source of it is NADPH or NADH, catalyzed by cytochrome P450 reductase or cytochrome P4B5 reductase. And when this second electron is also transferred to this to the complex, due to some further electronic rearrangement of electrons, one of the oxygen atoms of the oxygen molecule is left in form of oxygen, and the activated form of oxygen, the oxoid form of the complex, is generated, and this is the form which actually is involved in the oxidation reactions, resulting in incorporation of the second oxygen atom of the oxygen molecule into the substrate. No, so briefly, this is the cycle. And let's see the most important enzymes which are involved in hepatic enzymes, which are involved in transformation of drugs. So not only in the liver, but in the small intestine as well, the most important enzyme, cytochrome P450 enzyme, is the cytochrome P453A. Besides this, the 2D6 and 1A2 and 2A6 are the most important ones, which takes place in the metabolism, and we will see the examples of conversion of drugs, xenobiotics, by these three enzymes, not only in the liver, but in the small intestine as well. As for the drugs, I mean the marketing drugs, 40–50% of the marketing drugs still today is metabolized by the cytochrome P453A and the cytochrome P452D6 forms. And such a wide variety of the substrates expresses the very low specificity of these enzymes, obviously. That's why so many drugs can be metabolized by the same isoform of the enzyme. Now let's see some chemistry of these transformations. As I mentioned at the very beginning, the aromatic rings, aromatic moieties of the drugs are very common targets of cytochrome P450 oxidations, and as a result, the primary product is a epoxide, and this epoxide can further transformed, obviously, in a spontaneous reaction, results in the respective phenol, formally incorporation of one oxygen into the parent molecule. However, it can react with other enzymes or nucleophilic reactants. For example, epoxide hydrase and glutathione and some other macromolecules having active nucleophilic part, which can attack the electropositive carbon of the formed epoxide. The second important site of the cytochrome P450 oxidations are the double bonds. Oxidation of double bonds, according to the mechanism we have seen, also results in epoxides, aliphatic epoxides, and this reactive intermediate can further metabolize enzymatically or non-enzymatically, as we saw it before. The epoxide hydrolase can hydrolyze the compound, I mean the primary metabolite, to the respective vicinal diols, and however, it can also form a duct with glutathione and amino acids or any other macromolecules. These last reactions with macromolecules obviously modifies the primary structure of the macromolecule, which could lead to toxic effects. And that's another separate field of metabolic research, that what is the role of the metabolites of one particular drug in the physiology, in the biochemistry of the toxic side effects of the drug. Benzylic carbon atoms are also sensitive to hydroxylation. I mean, hydroxylate carbon attached to phenolic groups. For example, here is the tolbutamide metabolism, and which can result in a benzylic alcohol, and this benzylic alcohol afterwards can also further metabolized. Aliphatic hydroxylations could also happen, with several examples in the literature. For example, ibuprofen, pentobarbital. I mean, the metabolism of barbitals are well known since the very beginning of starting the metabolism studies. Ibuprofen metabolism, I'm mentioning here ibuprofen metabolism since one of the non-steroidal inflammatory drug we are investigating is ibuprofen, and ibuprofen has got several different sites, primary sites of metabolic transformation, among which the 1-hydroxy and the 2-hydroxy derivatives are the most important ones, and the 1-hydroxy derivative is further oxidized to the respective carboxylic acid under the physiological conditions. A very characteristic feature of drug metabolism is the oxidation, the cytochrome P450 oxidation of the carbon nitrogen, the carbon oxygen, and the carbon sulfur bonds. In such reactions in which a more electronegative atom is linked to carbon, cytochrome P50 oxidation results in a hemiacetal or analog of it, heteroanalog of it, which are unstable, as we know it from basic organic chemistry, and it results in the respective carbonyl, aldehydes or ketone, plus a small molecule as a side product. No, so this is, for example, among these reactions have got importance of the N-dealkylation reactions. For example, in case of imipramine, in which the carbon atom is hydroxylated according to the previously mentioned mechanism of action, and you see this is the unstable intermediate of the primary adduct, which losing formaldehyde and the norimipramine, which interestingly also very effective drug itself. The other transformation is the dealkylation of the ethers. The mechanism is very much the same. The carbon atom linked to the oxygen is oxidized by the cytochrome P450 enzymes. The hemiacetal, the formed hemiacetal spontaneously decomposes to the respective carbonyl and alcohol. So, for example, here is trimethoprim dealkylation according to this mechanism. However, we also know that codeine, which also a methyl ether, which also a methyl ether derivative, a methyl ether derivative of morphine, is also demethylated in the body, resulting in morphine as a metabolite of it. when we are having carbon sulfur system, the oxidation pattern would be somewhat more complex, since not only the dealkylation reactions could occur according to the previously mentioned mechanism. However, desulfuration of the thiones could happen. It's this conversion, unfortunately, is a toxic conversion, since elementary sulfur is released, which binds to the macromolecule. However, the thioethers could also oxidize to sulfoxide and sulfones, sometimes the sulfoxide or the sulfones being more active than the parent compounds. So these were the three systems, the CN, CO, and the CS systems, moieties, which are very common targets of cytochrome P450 oxidations. And let's see, what do we know about the phase 2? The phase 2 reactions are conjugation reactions, in which the parent compounds and or the phase 1 metabolites of the drugs are further transformed by conjugation with small endogenous molecules. The most common conjugation is the glucuronidation. Glucuronic acid can be found in the body in relatively large amount, and the enzyme UDP glucuronyl transferase is also expressed in high level, typically in association with the cytochrome P450 enzymes. Glucuronic acid can be bound to oxygen, nitrogen, sulfur, and even to carbon atoms. And the glucuronides are have got rather well water solubility and typically excreted from the body through the urine. There are some common diseases. I mean, typically in neonates, when the UDP glucuronyl transferase activity is rather low. So that's why some drugs are very toxic towards themselves. Obviously, not recommended, cannot be used. And the jodins, the neonatal jodins, may also be attributed to the inability of conjugation of bilirubin and metabolites of the bilirubin with glucuronic acids. As for the chemistry of the glucuronic acid conjugates, here are two well-known common examples. For example, conjugation of phenolic hydroxyl is a common transformation. A common feature of glucuronic acid conjugation results in phenolic ethers, which are stable and easily excreted from the body. However, there is another type of glucuronic acid conjugates, the ester conjugates. However, these ester conjugates are reactive esters, and due to the O-acyl migration of the substrate, the ibuprofen, for example, molecule can be migrate into the 2-hydroxyl or the 3-hydroxyl, leaving the anomeric hydroxyl free, and obviously it's in equilibrium in the cyclic and the open chain form of gluconic acid, and the open chain form of gluconic acid can be, for example, the final toxic agent of the molecule. However, being an active ester of the carboxyl glucuronic acid conjugates, nucleophilic part of macromolecules can also be modified, acylated by the substrate, in this respect with ibuprofen. However, it is, as I mentioned to you, there are some transformations which can be involved in development of toxic side effects of the molecules. The sulfate conjugation is the other important transformation, not only in the liver but in the small intestine as well. This occurs less frequently than does glucuronidation because of the inorganic sulfate level is typically lower than the glucuronic acid level. It's in mechanism of sulfate conjugation, PIPS, I mean phosphoadenosine 5' sulfate is the key intermediary, the co-substrate, which provides the active sulfate, and this active sulfate is transferred to the typically alcoholic or the phenolic hydroxy groups to the target molecules. Amino acid conjugation, as I mentioned to you, is a typical conjugation of aliphatic and mainly aromatic acids. For example, benzoic acid results in hippuric acid. I mean, conjugation of benzoic acid with glycine results in formation of hippuric acid. This is the historical sort of reaction, as I mentioned at the introduction. And the other example is aminoglycine conjugation of salicyluric acid, which also plays important part of elimination of salicylic acid from the body. Taurine, however, histidine, lysine, and alanine, serine conjugates also are important amino acid conjugates. However, glycine are the most common amino acid which involves in such a conjugation of xenobiotics. Glutathione conjugation is one of our main interests, not only from pharmacological point of view, however, from stereochemical point of view as well. And as for glutathione, is a tripeptide, is a tripeptide with an unusual acidamide bond. Namely, this is gamma glutamyl. So in this acidamide bond, it's not the alpha carboxylic acid of glutamate. However, the gamma carboxylic acid with glutamate is involved. So this is gamma glutamyl cysteanyl glycine, and it can be found in relatively high level in not only in almost all tissues, 5-10 millimolar level, and takes part very important part in the defense of the tissues and the cells against electrophilic metabolites or oxidative stress.
Hello, sir. Sir, you are not audible. Hello. Hello, sir.
Hello, I'm back again.
Okay, it's okay, sir.
This internet connection just
Yeah,
disconnected,
disturbing you. So
but
you
you
can
can.
further upload this slide.
Okay, so I continue it and I go back to a few slides.
Okay, okay, sir. That's
Just
no
a
problem.
minute. Once again, here we are. Okay, just a minute. Uh, I find it. I can't find it. I'm sorry. I got assistance.
Yes, that is very essential.
Yeah. Yeah. The entire screen. Yeah. Ano? Ako tsaka entire. Short naman. Entire screen. Okay.
Okay, sir. Now it
Okay.
is visible.
Can you hear me again?
Yes, yes, sir. I can
Okay.
hear you.
So, uh, did you hear this gluconic acid conjugate?
Yes, yes, sir.
Yes.
Yes, yes.
Sulfate conjugation.
Sulfate completed, sir.
Sulfate completed. Amino acid.
Yeah, amino acid remain.
Okay. So glutathione. Good. So glutathione conjugation is one of our main interests, since glutathione conjugation also occurs in the small intestine and in the liver as well. And the stereochemistry of glutathione conjugation is also a very challenging field from organic chemistry point of view. A glutathione level is relatively high. It falls into the 5-10 millimolar region in liver and in most of the organs, and has got a very special acid amide bound, and namely in the glutamic acid is involved in formation of an acid amide bond uh, by means of their gamma carboxyl group, not with the alpha. So the chemical name is that's gamma glutamyl cysteine glycine. Glutathione has got a relatively reactive soft nucleophilic sulfur, and most of the reactions of glutathione is associated with the reaction of this thiol function. The enzymes are also, I mean, the reactions, the glutathione conjugation reactions are catalyzed by the glutathione transferase enzymes. However, a lot of glutathione conjugation reactions proceeds spontaneously. Uh, uh, Most of the reactions, regardless it's nucleophilic substitution reactions like in this example, or nucleophilic addition reaction on activated double bonds, the first step of the reactions is attack of the nucleophilic sulfur atom towards the electrophilic carbon. This protonated sulfur is much more nucleophilic than the protonated one, and as a basic mechanism of the glutathione transferases is just to increase the portion of the protonated form of glutathione. As you can see, it has got two sites. One site for the binding glutathione, and the other site is binding the substrate, and the GSH with increased percent of its deprotonated form attack the electrophilic carbon, resulting in the glutathione conjugates. The glutathione conjugates themselves, as a primary adduct, undergo further metabolism, as you can see. In the first step, the glutamate is hydrolyzed. Afterwards, the glycine molecule is lost, and the cysteine adduct is left behind, which is acetylated, and the acetylated form of the cysteinyl adduct, called mercapturic acid conjugates, are the typical metabolites which can be identified and quantitated in the urine of the humans or in our experiments in the experimental animals. Finally, the two other, the acetyl conjugation and the methylation. As I mentioned, these two transformations increase the hydrophobic character of the substrates, decrease solubility. In this case, the acetyl moiety is activated through their thiocoenzyme A esters, and this activated acetylated acetyl group is transferred by nucleophilic sites, typically for nitrogen and oxygen molecules of the molecules of the substrate molecules. The methyl conjugation, this is a minor conjugation method. The activated methyl substrate, co-substrate, is S-adenosyl methionine. So it means methionine is the precursor of it. Energy required for these formations. And this is the co-substrate of Materialtransferases, which can transfer the methyl group to oxygen, for example, in case of phenol O methyltransferase, or nitrogen, and for example, N-methyltransferases, and some other examples are also not as important as these two, for example, the sulfur methyltransferases as well. Here are two examples of methyltransferase reactions, and it's important to tell that these metabolic transformations can metabolically transform not only the exogenous, however, the endogenous substances as well. For example, norepinephrine can be methylated by means of the catechol methyltransferase at one of its phenolic hydroxyl, and the paranitro methyltransferase to result in formation of epinephrine. And let me, in the last 4 minutes, tell something about our research associated with hepatic and small intestinal transformation of of foreign substances. We originally started our research with the model substance nitrophenol and investigated how the metabolic feature of nitrophenol is changed in animal experiments as a result of hyperglycemia. Hyperglycemia and other diseases can modify the enzymes, the activity of the enzymes, the metabolic enzymes, plus the activity of the transporters, which sometimes involved in transformation of the compounds through the membranes, and accordingly, the pharmacokinetics of the drugs we have investigated under control and hyperglycemic conditions have systematically and characteristically changed. For example, here is another piece of work of us. I'm sorry, I forgot to translate the title from Hungarian, but you might understand that this is about the capsaicinoids of intestinal elimination. I mean absorption of capsaicin and dehydrocapsaicin in our system. And what is also important that the metabolites, the capsaicin metabolites, and not only the capsaicin metabolites, but the ibuprofen metabolites and the nitrophenol and paracetamol metabolites, after entering the small intestine epithelial cells, it can be re-excreted to the lumen, and in the lumen you could see, for example, in case of capsaicin and dehydrocapsaicins, the glucuronide gluconic acid conjugates of capsaicin and dehydrocapsaicin was continuously increased in the lumen. I mean, the capsaicin absorbed, and in parallel, we observed that the gluconic acid conjugates of capsaicin and dehydrocapsaicin is re-excreted to the lumen. As far as I can see, my time is over. So once again, thank you very much. Thank you so much for the possibility to tell you some really introductory lecture talk on drug metabolism. As you can see, this field of research might be interesting not only from pharmacological, medicinal chemistry point of view, however, from organic chemistry point of view, needs different skills. For example, needs skill in organic chemistry, analytical chemistry. So our pharmacy students are frequently join the department as research students, since this is a topic they can make use after they got their pharmacy diploma. Once again, thank you so much. Thank you very, very much for your invitation, and not to mention the patience of listening to my lecture.
Thank you very much, sir. You nicely explained the lecture related to drug metabolism and the different phases of drug metabolism. Actually, that goes through that is essential for any drug to metabolize inside the body. That phase 1 and phase 2. And you nicely explained about the phase 1 metabolism pathway, different type like oxidation, reduction, dealkylation, hydroxylation, and phase 2 metabolism, glucuronide conjugation, And you also explained different cytochrome P4 types. So, sir, really, we enjoyed a lot and got a knowledge related to this drug metabolism. So, thank you very much, sir, for joining with us. Sir, now our director, sir, want to share some views. Hello, sir. Am I audible, sir? Dr. Pal, am I audible? सर आपका माइक बंद है सर आपका माइक बंद
Thank you very much to all for the organizing this very nice interactive session. Sir, we are going to organize these types of the ISFCP dialogue series for the benefit of the students of the pharmaceutical sciences, those who are working in the different field. And this is the recorded as well as we run this whole of the lecture through the YouTube channel, sir. And ISF College of Pharmacy, one of the premier institute in this India, and we are running a postgraduate program in 9 branches, as well as we have the one of the specialized lab. This is the zebrafish lab, cardiovascular lab, and we have the excellent facilities in the pharmacology. We are running one of the animal house breeding and trading facilities by government of India. So near about 20 of the university, they are just visiting our institute and they are purchasing the animal from this institute. We are running one of the testing laboratory of this is approved by the NAVL. So this institute is the well organized and well known for the research, sir. And I hope that in future, if we have some of the collaborative work with India, so you're welcome, sir, because you know that you have the lot of the grant facilities at the up to the mark. So if you need any human resource as well as you need some of the assistance from my side, so my institute as well myself, we will be the very happy to provide the services to you. So thank you very much from core of my heart to accept our invitation, and you have delivered a excellent talk. Thank you very much, sir. So Dr. Vivek ji,
yes
I
sir
think connection problem is there net. So thank you.
yeah sir
And
I think internet problem is there
okay, okay.
they disconnect now I'll connect again
I'm very sorry. These troubles. Typically, there is
it's
no
okay sir it's
troubles in
okay
the. But
Thank
now,
you, sir. No problem.
and it will be very important. Several troubles occur. So
Okay,
thank you, professors. Thank you so much, and I really appreciate the offer to develop a joint research activity or publication activity. If you don't mind, I send you some recent publications, both of you, and
yes sure
afterwards
sir
by phone or by correspondence, we can set the further details. Thank you
sure,
again.
sir.
yes sir
Thank
sir
you. Thank you
Thank
very
you.
much.
we thank you so we hope that we
Goodbye.
do some collaborative work in future we do some collaborative work in future sir
Just a minute. I did not mention in the lecture, but we have a HPSCMS facility. We have an animal
oh
laboratory, so we can make the surgery in the institute, inside the institute, and in the next door lab we operating HPSCMS. So we collect samples from from bile and from the small intestine perfusates, and we analyzing these biological samples by HPSC and HPSCMS. So in this respect, we can have flexibility in analytical works and animal works, whatever.
Definitely, sir. Definitely. Thank you, sir.
Thank you
thank you very
again,
much sir
and
thank you
goodbye.
thank you
Goodbye. Thank
goodbye
you.
sir